Welcome to part two. I'm really glad you guys could be here this afternoon, and we will talk through kind of an interesting problem in proteomics. How can you judge which PTMs you should look for in a DDA proteome? In this case, we are dealing with eye lens material from a person who was 93 years old at the time of donation. Maybe this was surgery, maybe it was postmortem. I'm not actually sure in this case. But the eye lens of a human is built out of proteins at a really early stage, and they're basically kept with you your entire life. So if you're asking what kind of damage a protein racks up due to the environment, you do quite well with the crystallins in the eyes because they've been exposed for decades to environmental effects. This is, I think, the second lens paper in a row out of this group. Some folks I used to work with, actually, who are working with aging and cataractus lenses, trying to understand what are the spontaneous PTMs that occur in these proteins just from years and years of exposure. The data set itself spans quite a few different people, and it includes multiple fractions for each person. So I've chosen the 93-year-old because that was the oldest person in their sample, and I thought it would have kind of interesting PTMs to look at. And I've picked out 3 files from the urea soluble fraction, the USF, from this person, because I know these 3 files from the separation include lots and lots of interesting IDs. So I thought it'd be a good place to start. If you're interested in the science that led to these proteomes, I've included the PDFs on the disk. One of the things I want to mention here is that the human proteome database, the reference proteome from Uniprot, is quite large when you include the unreviewed sequences and when you include the Swiss-Prot proteoform, the Swiss-Prot isoforms as well. So as you can see, 107 megs of FASTA takes a bit of time to do searching through. In this case, I've already done a basic database search of these DDA experiments, and I decided that I would start us not with the full proteome, but with just the proteins that you can identify in an ordinary search of these raw files. So we were discussing earlier that some of the workflows for FragPipe can export a protein.fas file, and that FAS file is a FASTA that contains just proteins for which some peptides have been identified. So it's a quick way to get a subset database that you can work with. And in this case, we'll only care about modifications to proteins that could otherwise be identified. So it should cut out a lot of our search space. During the break, I have installed FragPipe 23.1, so I should be a little more current. And if you see differences between your display and mine, then that's worth noting. But I've now got all the basic tools installed, MSFragger, IonQuant, and DiaTracer. Diane is the same version as before. I want to note, by the way, that this is not the current version of Diane. If you were to download Diane from their website, you would end up getting version 2.0 something at this point. So the version shipped with FragPipe, I think, is quite specialized to answer these particular calls that get made to the software. It's not necessarily something where you can just sub in version 2 and everything would work fine. All right, and Python is installed. That's all in a good place. So for workflow, I would like to try one of the workflows that are most distinctive to FragPipe. FragPipe was created to support, MSFragger was created to drive open searching. And this is not what we would consider a standard way to go about searching for PTMs in proteomics. Open PTM search is what you do when you don't know what mass shift is found on what amino acids. That's quite different than like a phospho searching. So if I were doing a phosphoproteome, I would probably look for plus 80 because that's the mass change induced by phosphorylation, and I would look for it on serine, threonine, and tyrosine because that's the 3 places it can be found. I might also look for minus 18. Why would I look for minus 18 on that? So when the phosphoserine kicks off phosphorylation, it spontaneously, it may not do so in a mechanism that recreates serine. Instead, you lose water from this. You lose phosphoric acid instead of the phosphorylation. So instead of losing 80 from a phosphoserine, you could lose 90. Instead of gaining 80 to be phosphorylated, you might have minus 18 to reflect the spontaneous elimination of the phosphorylation. But these are the kinds of searches that database search has already been very good at since 1995, basically. That was when it was first published, how to go about doing variable modification searching. You specify an amino acid on which this change can happen, what the mass shift is, maybe you indicate what its unimod identifier is, and then off you go and you're searching, you're good. Open search is a different animal. Open search is intended to say, I don't know how much mass has been added or removed by this set of PTMs, and I don't know what amino acid it's attached to either. That's pretty specialized. It is also possible to say, I'm interested in all peptides that spontaneously gain 32, for example. You could make a much more specialized open search. So you could say, I know the mass, but I don't know where it should be. So there are several options made available. In this case, we're going to use the wild and woolly option. We're going to use the blind modification search, which is what we would have called it in the sequence tagging days. We will say some mass that could range considerably in size is found at one location on a given peptide. So let's go ahead and set up for our open search. Now that I'm in version 23, I no longer have to click load this option. That's good. But now I can add the files for this particular run. Okay, so we don't want to do the part A changes. We want to do part B. And in this case, they're already expanded out because we didn't zip these in the first place. They're just raw files. And this is a nice strength of working with FragPipe that is capable of reading raw data as is. You do not need to convert your timsTOF data into an MZML before running a search on it. Now, if you're running something like a SciX triple TOF, I believe it is still required that you convert to an MZML before searching within FragPipe. But at least for the timsTOF and thermoset, you can get by with just giving it the data as is. Oh, you can actually see it down here. It'll search an MZML, an MZXML, which is even older, an MGF format, which I don't think you need to worry about too much. That's what you would use for mascot searching. The MZBin, I think, is the mass shift recalibrated format that it creates. Thermo raw or Bruker.d files. So that's all fine. We're going to use these three raw files as our input. In this case, I am not going to be feeding this to FragPipe Analyst downstream. So we're not going to be asking what proteins or what modifications differ among these three files. We're going to just treat them as one big lump of file. So I'm not going to do anything about specifying experiment or bioreplicate here. DIA sudo ms2 is turned off. Database. We need to change our database because we're no longer dealing with yeast with human protein spiked in. We need to back out and go into discovery of an iLens. Here I'm going to specify the subset database. So we have just the proteins that have been previously identified in these data for just the 93-year-old donor. And you'll note that it says decoys. We've already added the reversed version of each protein sequence as well. All right, so I can now click on select. And just as a double check, it's always worthwhile that you check that FragPipe reports that 50% of the files, 50% of the sequence database entries are decoys. If they're not 50%, you have to take that into account when you compute false discovery rates. And if you simply reverse every protein entry, it should be 50%. It's also pretty obvious that this is a much, much more compact database than we used earlier. Earlier, we started with 6,000 yeast proteins, added in another 50, and then doubled it all, right? Here we have 296 real proteins that are showing up in decoyed form as well. So that will cut into our search time quite a lot. All right. Now, MS-Fragger should, because we selected the workflow was open search, already be set up for wide tolerance searching. And we start with the precursor mass tolerance. This is obviously a little bit crazy. We have a mass tolerance for how accurate the peptide mass should be, to be anywhere from minus 150 Daltons to plus 500 Daltons. That's a pretty big window. Now, if you're dealing with something like complex glycans stuck to a peptide, you probably will need to make sure that the complex glycan structure you're looking for fits within that window. So if it's larger than 500 Daltons, you have to expand that range out a bit. But if you were doing that, you would use that workflow. So hopefully that would take care of it for you. Our fragment tolerance is still set to 20. Again, it's going to try to refine that along the way. We're still going to act like this is trypsin. If you felt that the sample was really dominated by just a few proteins, like you were dealing with serum or plasma or something, then you might choose to do semi-tryptic searching here. It would actually help you out a lot. But semi-tryptic in combination with open search is quite a heavy burden. So here we're going to just do the open search and act like everything is a very well-behaved tryptic peptide. Okay. As we move further, we see there are still a couple variable modifications in effect. The oxidation of methionine, the acetylation of N-termini is still there. And we still have cysteine here. Remember, if you didn't use chlorocetamide or iodocetamide, you need to change away from 57. And we're not doing the specific mass offsets where we have particular mass shifts that we know in advance are going to be present in the sample. Modifications are off. There's a bit of stuff down here that we would pay attention to as well. Do you see this localized mass shift feature? That's pretty important because here it's more important. It's important to us that we say not just this is the peptide sequence plus this amount of mass, but rather where in the peptide is our best guess to place that mass. So the fragment ions may give us the ability to pin that down quite well, or maybe not. But we want to use that information if it's present, and localizing the mass shift matters for that. Are there other things that I want to discuss here? I don't think so. But again, we're not actually changing anything. We're just using the bog standard open modification search layout for this. Validation is just the same as before, except now we're not running MS Booster. Instead, we're running Crystal C up front. And I believe Percolator will be handling our separation of good IDs from bad still. What else do I want to note here? Yeah, there are a fair number of pieces in here. Obviously, the options continue running all the way down here. Is this where it does the generate subset database thing as well? I was trying to find that. Well, I'm not finding it at the moment. It's okay. All right, so the validation thing, we're leaving everything as is. Suddenly, the PTM Shepherd options are on. You remember when we did this search for the DDA and DIA cohort versus cohort stuff, PTMs was disabled, but now the PTM Shepherd software is turned on. So Crystal C and PTM Shepherd are going to play complementary roles here. What if you had a semi-tryptic peptide in a tryptic peptide search? Does everyone know what a semi-tryptic peptide is? Oh, okay, so to be careful about this, a semi-tryptic peptide states that one end of the peptide, maybe it's the N-terminus, maybe it's the C-terminus, one or the other is tryptic, but the other side doesn't conform to where trypsin should cut. So imagine that you had a signal peptide that got lopped off of the N-terminus of a protein, and that didn't end in R or K. Well, that first peptide would look like a semi-tryptic peptide because the N-terminus does not correspond to where trypsin would cut, but the C-terminus does. You could have, you might be running your source a little hot. As a result, you might get some large peptides getting cut into pieces as they go through the source, and you end up with what looks like a semi-tryptic peptide. It's not because another crazy protease was at work. In this case, that's an electrical phenomenon causing your peptide to get broken up. So being able to find those, being able to account for those is handled by Crystal C. It's able to recognize semi-tryptic peptides that show up in a tryptic search. And PTM Shepherd is very useful for looking at patterns of behavior. And this is going to be a very useful report for us in evaluating the open search that comes back. So I just wanted to note that these are showing up when the others are not. Glycos, still turned off. Quantification, turned off. We're not going to attempt to quantify at the same time as we look for patterns of modification. We're not doing isobaric quantitation. We're not generating a spectral library. Not doing a DIA quant. No skyline. So we're basically back to the Run tab just as before. I now want to create an output directory. This one will be for part B, so I'll navigate into there. Training 2, open search. It's a fine name, we'll go with it. Training 2, open search. And I want to delete temporary files, and yeah, I think that's basically it. Now I'm going to go ahead and click run, so this goes ahead to launch. So let's talk about search space for a minute. I think that it's a fundamental concept, but it bears talking about it. The search space is the set of peptides and decorations that must be considered in explaining a given MSMS. Okay, so we often talk about the sequence database as the search space, and it's a start. So if I have one sequence in my database or 100,000 sequences in my database, that's obviously got a pretty big difference in how many comparisons I must make to a given spectrum. How about the effect of precursor mass tolerance? If I allow for a 10 ppm peptide mass error, then I make far fewer comparisons than if I have a 2 Dalton window, right? I mean, that's 10 ppm versus 2 Daltons. It's a pinprick versus a massive space. So, so the more tightly precursor tolerance is set, the fewer the peptides you end up comparing to each spectrum. It is still running. That's good. Oh, we see their mass errors are a little higher here. We're in the range of almost 4 ppm off. So it's good to get those corrected for doing the search. Okay, what about trypsin? When we do searches in FragPipe, the default behavior is almost always look for fully tryptic peptides, those that conform to trypsin specificity on both ends. But how does the search space change if we tell it to look for semi-tryptic peptides? The answer is more than an order of magnitude. More than 10 times as many sequences are going to get compared to every spectrum. So that's just a small subset of the things that apply to search space. When we start adding on post-translational modifications, our search space can really bloom into something entirely other. If you include just serine, threonine, and tyrosine modifications, that's already more than doubled the number of peptides that get compared to every spectrum. And the reason is we have an exponential growth in how many different ways a given peptide gets compared to spectra when we add PTMs into play. And assuredly, doing an open search is going to build onto that search space quite a lot. Almost any peptide sequence can be compared to almost any MSMS if you have the right PTM shift added to it or subtracted from it. So open search is in large part throwing out the value of the precursor mass filter in doing these identifications of MSMS scans. All right, so we're already past the initial database search, and now Crystal C is doing its thing to help us screen out some phenomena that could look like PTMs that aren't really PTMs at all. So let's imagine that I have a peptide that has a modification of 156 on its C terminus, but I happen to know that the next letter in the amino acid sequence is arginine. That would look like a modification of 156, but 156 is the mass of arginine. So there are lots of things that can cause an apparent PTM, but they're not really PTMs at all. What about the replacement of one amino acid for another? What if I had a proline but I saw that it had a mass shift of 16 on it. Proline has a mass of 97. A mass shift of 16 then would cause its mass to become 97 plus 16. 97 plus 16. It's after lunch. We're all struggling. 113. 100. Yes. So proline, if you add 16 mass units to it, is 113. So if you tell your software that I might have mutations in these data, and it says I see a whole lot of cases where proline has been replaced by leucine, that's one plausible explanation for why you gained 16 on a proline. However, the biochemists and the cell biologists will tell you that's just silly. The sample you were analyzing had a bunch of collagen in it. Collagen has a lot of hydroxyproline. Proline oxidized has the same mass as leucine. So if you tell the software, I'm looking for only this type of modification, you got to be careful that there aren't other explanations for that same mass shift. Okay? If you have 156 added to the C-terminus and the next residue is arginine, that's probably a safer explanation than an actual modification of that mass. All right. Well, that was a really fast search, wasn't it? Just 3.9 minutes. Did yours all finish just fine?
Yeah.
Oh, but you have a question. Yes.
Yeah, I have a question about how do we solve this. If you say this mass shift can be not very sensitive, how do we know which one is which?
That's great. That's great. The rule I most typically use for this, how do you choose among different hypotheses that could all explain a mass shift? My answer is the boring one is correct. If you see a gain of 22 on a peptide, that's sodium. You have a little bit of sodium in your sample, and plus 22, or is it plus 20, plus 23? Oh my goodness, I should have this up. What? Oh, we're okay. It's Friday afternoon. It's setting, and I'm afraid. So I'm going to pull this one. 23. Ah, see? Wait, is that the atomic mass or the mass shift?
That's the atomic one.
Ah, okay. So the 23 replaces a hydrogen, or a proton, I should say. Therefore, yeah, plus 22 is the gain, reflecting an addition of a sodium ion instead of a proton. Yeah. Whenever you have competing explanations for why the mass shift is seen, the most trivial, most boring one that prevents you from getting a Nature Biotechnology paper, that's the right one. All too often, we see publications in very major journals by very major groups that a very careful bioinformaticist can pick apart by saying, this mass shift is... this mass shift that they're seeing is easily explained by this trivial phenomenon, not what they've claimed, which is that they've validated a rare mutation in this particular tumor or whatever. This stuff happens all the time. So yes, finding the most boring possible reason to explain your mass shifts is not gratifying, but it will protect you from making a silly statement in press.
Don't look for zebras until you rule out horses, yeah?
Sorry.
Don't look for zebras until you rule out horses.
Don't look for zebras until you rule out horses. I really like that.
If you hear popping hooves, look for horses before you think seagulls.
I like that. Okay. Well, our search has run. How about we figure out how to interpret it? Well, what resulted when we ran this search? I dumped these results into training to open search. And I see I have my list of stuff here. I've got things like the protein FAST file. This is the subset database created from our subset database. I still have my log file so I can remember how this search was run. But I'd like to spend a little time with the PTM Shepherd output. Now, I hope you'll bear with me. It's been a moment since I created this tutorial, so I got to remind myself of which facts appear in which places. So don't do bioinformatics in Excel unless you really, really need to. So let us now start with the simple version of the table. the global modification summary. Thank you for chiming at me. Okay, so what we have is not a PSM by PSM appraisal of the sample. What we have here is an overall examination of how popular particular mass shifts are. So we start with the base case. Adding a mass shift of 0 shows up in plenty of different places. 31% of the PSMs overall included that. Look at the number of the fraction of these deamidations that occurred. This is a case where a mass shift of 1 occurred between what we thought the mass should be based on the peptide and what was observed. So deamidation in this case was a really common outcome. Dihydroxy. Here we have a gain of 32. So does anyone have an idea of what that 32 is going to represent? I mean, the chemistry is spelled out right here. It's a dihydroxy, but what is that?
Hydroxy is you add an alcohol group, and if you replace a proton with that, then you get plus 16.
Yeah, so if you had an OH that got added onto that replaced an H on a methionine, we call that an oxidation. If it happened twice, you'd have a dioxidation. So a dihydroxy is what this is. So we're going to see, does this tell us what amino acid that occurs on?
No.
I don't see that information yet. Okay, oxidation or hydroxylation, carbamylation. Does anyone know what carbamylation is? He's stopping chemistry now. Well, occasionally you have an old mixture of urea that you're using for denaturing your proteins. And if that happens, you can modify peptides at their C termini by this mass shift of 43. That's carbamylation. But then we also have a couple of things that we can't really explain easily. An unannotated mass shift of minus 43 or of plus 50. Got some weird stuff going on. So we're going to want to explain these. And of course, there's this big fall off in how many PSMs incorporate that PTM as well. So at some point, we're going to need to draw the line and say anything past this, I just can't care. It's a minority PTM that occurs in 5 spectra. I'm not going to go chasing it. Okay? That, I think, is one of the more difficult lessons to convey out of these data. And I'm going to try to walk us through how we would interpret which PTMs we declare are real as we move into the next slide, the next spreadsheet. So let's close the global mod summary and instead look at the global profile. Okay, now, can David remember every aspect of this? No, but he can get us started at least. Remember that we start, do you see this peak apex column? You should read that as, what is my best guess as to the true mass of this mass shift? So the peak apex of 0.00002 is to say no mass shift. And we see that that's really common. We don't try to localize it because it's no change. There's no way to localize no change. All right, as we move to the next line, however, we get to deamidation. You remember we saw a very large number of PSMs that could be made if we allowed for a 1 Dalton mass error. And that's included in ordinary searching, actually. But what if the problem here is not that we chose the wrong isotope from the MS scan, but rather that at some point in the database sequence, we need to make a little addition of 1 mass unit in order to make this sequence match the fragment ions. That's different, isn't it? So in this case, is there a reason why the chemistry of peptides being in the environment for a very long time could lead to a gain of 1 Dalton? Let's get some hints on how this could happen. We come over here and see that there's not much sighting of that at the N-terminus of the peptide. So we're not gaining 1 Dalton on the N-terminus. That's not common here. The most prominent amino acid in which this happens is asparagine. It's enriched many-fold past what you would see by random effect. And the second most common place we see it is in glutamine. Are we any closer to guessing why we would gain a Dalton here?
Protonation on the acid groups.
No. This is bog standard deamidation. Within a peptide, if you have something like an asparagine followed by a glycine, you can end up with some weird chemistry between the side chain of asparagine and the following peptide bond. And one of the things that can happen is that asparagine in solution can turn itself into aspartate. So the difference between asparagine and aspartate is 1 Dalton. Similarly, the difference between glutamine and glutamate is 1 Dalton. And this comes about when you get some weird interactions between the side chain of one of those 2 amino acids and the next peptide bond. In some cases, you can get weird changes of the peptide backbone so that the backbone no longer continues NCC, NCC, NCC. Instead, the rest of the peptide is hooked onto the side chain of asparagine, or of aspartate. So you can get some really weird stuff from it, but the upshot, I mean, you didn't think it would be a big story. It's 1 Dalton, right? But here it is. 1 Dalton can occur because asparagine and glutamine are doing silly things. Okay. So the gain of 1 Dalton could come about because of making a mistake of picking the monoisotope in the MS1 scan, or it can come about in the case of an N or Q that's getting adventurous. Either of those will take place. All right, now we saw that 32 was really popular. Here it is again, dihydroxy. Under what conditions do we see dihydroxy getting showing up in a lot of popular ways? Here we see that W is very, very much overexpressed, is very enriched among those peptides that get dihydroxy. So tell me about tryptophan. What do we know about it? Okay, I can tell you that every Thanksgiving, Americans eat a whole lot of turkey and then they get really sleepy, and that's the effect of tryptophan. Okay, but what about the amino acid? What is it about the amino acid that's distinctive?
It's big, it's aromatic.
Big and aromatic. Great, that is a wonderful place to start. Tryptophan is also a bit reactive over time, and it's not content to limit itself to just a single oxidation. It can oxidize multiple times. If you expose tryptophan to air for 90 years, it's got a pretty good shot of double oxidation. That's why this is dihydroxy. Likewise, we have proline over here. Proline is kind of a squirrely amino acid when you get down to it. It can always get up to something. And dihydroxy modifications are one of the possible ways. One of the things you should know is that your cells use a lot of this ECM, extracellular matrix, to kind of glue themselves to their neighbors and to communicate with each other. And that stuff is a large amount of collagen protein. Collagen has a huge amount of modified proline. So I mentioned the weird case of proline having one oxidation looking a lot like leucine. Similarly, we can see double hydroxy modifications here. Okay, we move on to the next one, single oxidation. All right, so we know that oxidation happens to methionine, right? But hold on a second, what's going on here? Single oxidation was most overrepresented in tryptophan. So tryptophan's playing this sneaky trick on us that it might be doubly oxidized or it might be singly oxidized. Who knows? It may feel that way today and a different way tomorrow. Similarly, tyrosine. Tyrosine can carry this single oxidation. And proline. Why didn't methionine show up here?
Because this is a list of unexpected.
Very good. Yes.
And we predefined the methionine oxidation in the fixed modification.
That's right. When we configured our MS Fragger search, under MS Fragger, we had listed methionine as spontaneously oxidizable as a variable modification. Variable modifications are handled separately from unknown modifications. So these unknown modifications are all the cases that are not included in the variable set. Okay, so that's valuable to know. We see that things other than methionine can also get adventurous and gain oxidation. What about carbamylation? What did we learn about carbamylation? Well, we knew that we had some old urea on hand, and in this case, suddenly column M has a value. These are N-terminal proclivities, basically. So if the modification is not happening on a particular chemistry of side chain, it may be happening on the terminus specifically, because they all have a peptide N-terminus irrespective of what side chain is there. So when you see a large number in column M in this N-terminal localization, it means there is at least some hint that this thing is attributed to the N-terminus, not a side chain on the body of the peptide. So carbamylation of N-termini is quite likely. So yeah, that's where you would look for your plus 43s. I think before I said it happened on the C-termini, I'm sorry. It happens on the N-terminus, obviously. And also, we see that lysine has a very high enrichment score. Why would you expect a commonality between the modifications that happen to an N-terminus and the modifications that happen to a lysine?
Both are primary.
Yeah, the chemistry is quite the same. In the case of lysine, you just have a long alkyl chain with an N on the end of it. In the case of an N-terminus, it's not on a long alkyl chain. It's just the end of an NCC backbone. But both of them are just a primary amine, and both of them can take on the same modification. So the carbamylation might happen at an N-terminus, or it might happen at a lysine. When we look at the D here, do we think that this aspartic acid is a strong signal or not? And here I would point to the amino acid 2 enrichment score. Here it's only a 1.6. If that number is above 1, well, it happened more than you'd expect by random chance. But 1.6 is not a lot more. So there's a much better claim here that the lysine is strongly overrepresented among things that gain the carbamylation. There's a much weaker proclivity for the aspartic acid to be associated with that same mass shift. Does that make sense? We can talk about what these log odds ratios represent, but I've forgotten the specifics of it, I'm afraid. But I believe that a 1.0 means happens about as much as you'd expect by random chance. Okay. Now, we had some other business going on. Here we had a mass shift of minus 50, or was it plus 50? Sorry. Oh, this is the plus 50. The other one's a minus 43. Where did that happen? The plus 50 has no N-terminal localization, but it does have some affinity for phenylalanine. Did I say that? Yeah, that's phenylalanine. Had a brief moment where I worried I suddenly wasn't, I was thinking P was phenylalanine in my head. Long tutorials get your brain scrambled. How could I check on a website, let's say, what amino acids, what is a possible chemical explanation for why phenylalanine would gain 50? Anyone know of a website that can help me? I'm going to put us on Unimod, because Unimod is your best friend if you're doing PTM searching. I'd always ask you to log in, but feel free to say log in as guest. I still haven't created an account on there in all these years. It's embarrassing. Now, they said that the mass shift was 49.9. I'm going to do a quick searchy poo for that and see what I hit. All right, so I see 49.97 would be explainable by a leucine or isoleucine becoming a tyrosine. Does that correspond with what we saw? No. It said phenylalanine is associated with this. Okay, so we've got nothing here. Unimod is showing us no mass shifts that are particularly associated with phenylalanine and a gain of 49.9. What can we do with that? Well, it is possible to look through all the PSMs and ask on which peptides did I see this mass shift of plus 50 and how many were for phenylalanine. That's kind of, we're seeing a digest of that, but you could look through the specific instances of it. We could then ask, honestly, this is the thing we would need to know. do we have reason to believe that multiple mass shifts exist on this peptide that together add up to 50? Isn't that messy? So what if you had a pair of plus 16s, that's 32, and then you had an additional, what, 28? 18 in addition. So you could have a watery amino acid and a double oxidation somewhere on the peptide. That would add up to 50 as well. So remember that the search space said we can add one mass of indeterminate size to any one amino acid. That's not a limitation our peptides are under. They may have multiple modifications that add up to that mass as well. And if the plus 50 we saw is coming from a protein with oodles and oodles of copies, then it may just be giving us a lot of evidence over and over again for the same sequence being modified in this way. Okay.
So,
yes, sir.
I was wondering, because if you were to look at the data, right?
Yes.
You would be able to spot the localization of a mass shift based on wrong distances between adjacent Y ion or B ions. Yeah?
Yes.
And so, presumably, this identification of a plus 50 modification on a phenylalanine is based on some sort of mass shift between a phenylalanine and the previous amino acid.
Yes.
But that mass shift would not have been 50, right?
Why not?
Well, because unless all of the modifications happen on the phenylalanine, right? If you have, say, you have a chain and you have multiple modifications, but not on the same amino acid residue,
yeah,
then the shift in mass that you see when you remove the phenylalanine versus having it there, it's not going to be, that offset is not going to be 50 because you still have a fragment and another modification elsewhere, right? So
okay. I think Chris is getting to a problem that I've kind of alluded to vaguely, but we really need to talk about in more detail. And that is localization. How does the software pin down this location?
Yeah, so it does that based on when, at what point in your known sequence you see a wrong mass shift between two adjacent Y ions or B ions.
Okay, so in this case, we look at this particular record for just this row. We see that phenylalanine has an enrichment score of 4.5, which is kind of marginal, and has a PSM count of 20. So this is a summary coming from 20 tandem mass spectra that bear testimony that phenylalanine is the place where where the mass shift is seen. Do all of them localize it to phenylalanine and only phenylalanine? No. Do we want to go digging into it? Well, I mean, one can.
In this case, it's not so bad.
Yeah. The files in PTM Shepherd output, they are summaries at a high level. Like I said, that one was summarizing 20 tandem mass spectra for just that one amino acid. So we can go into the modification data and dig through this. Now, do I need to do this on the PSM or the peptide list? Let's see where we get with the peptide list for a start. All right. Now we are looking at peptides. Does this show the modifications within the peptides? I don't think it does. This is not showing modifications to the peptides, I think. Oh, here it is over here. Assigned and observed. Very good. Okay, so acetyl. In the other table, those were called unannotated. So let's see if I can do a quick search for unannotated. Helpful. Okay. If I just skip down here.
It should also be possible to, like, if you search column M for F, then you'll find things that are localized to phenylalanine, right?
The column M is only giving.
So it gives the 3C whatever is a modification on cysteine that is in position 4.
Right. Okay, so if I were to look within column M, search by column. What if I look for that 49.9? That might be the better way. Oh my goodness, look at this mess. All right, well, this is, we're getting into a deep dive. Look at the rationalization for this mass shift. Cation of nickel, formylation, trioxidation, and three things we can't even describe. That's a nuts localization. So yes, it might be possible to find those 49.9s if I'm very, very crafty. To find them solo could be difficult. I don't know. I'm not going to dig into it at the moment, but it is possible then to see what is the spectral evidence that this PTM exists. But I'm kind of dodging around an important question here. Are we going to submit a manuscript to Nature Biotechnology saying we've discovered these amazing mass shifts on eye lenses? Look at this open search. I strongly, strongly urge you not to do this. People have tried and reviewers like me have shot them down. What you should instead do is try to build the best conventional database search you can to take these mass shifts into consideration. And that will give you a much more controlled error rate as you build a trustworthy set of identifications. So what we're going to do is rerun our database search on these data. I come back to FragPipe, but this time I'm not going to run the open search. Instead, I want to run I think I want to run a conventional basic search. I believe in the earlier version this is called the default workflow, but you can do what you like on that. We're using the same database as before, just because it will save time. We could search the entire human proteome with this. But on this page, we're going to do a lot of decoration. So I hope you'll bear with me as we try to work through how to populate this modifications table with the information from this one. So let's start at the top. We don't need to add any modification to represent 0. That is the default search anyway. That's already there. We want to add deamidation to our search. It is the single most common type and we need to of PTM, and we need to add it on N and on Q. So let's see if that's already in place for us. All right. If I look through, I'm going to look for +1 mass shift. I'm not seeing it. So I'm going to overwrite phosphorylation here. So let's do this. All right. I want to specify that either N or Q can gain 1 mass unit. I'm going to use Unimod to help me out here because Unimod has all these helpful annotations in it. I'm going to do a quick search for deamidation. Here it is. And we see its monoisotopic mass is 0.984. Copying that, and copying it into here. All right. Now, is the canonical mass of deamidation the same as what we saw in our open search? It says it should be 985. Unimod says it should be 984. That's pretty spot on as these things go. Yeah, definitely close enough. So we've now added 1 modification on N or Q that was represented in our open search. That's cool. Now, next up, dihydroxy. This was not included in the defaults because it is it's a pair of oxidations, not a single oxidation. So let's now get the mass for that. I'll do a quick search for dihydroxy. Here we go. Second row. So dihydroxy has a mass of 31.989. Does that agree with what we saw here? 31.9904, 31.989. That's pretty close. pretty close. I'm going to go ahead and write that into our configuration. But where is it happening? Dihydroxy is happening on W and P. W and P. So I'll just write over the next one. W and P are now allowed to gain 31.989. All right. So we've now accounted for the top 3 rows. Let's look at the next one. Remember, this is oxidation. Oxidation is already in our search, but we need to change it, don't we? Because oxidation is not limited to methionine. So we need to add W and Y, and if we're feeling very adventurous, P as well. So let's just do it. P, W, Y, and P. Oops, I'm hitting the wrong button here. Let's go up to the top line here. We're just going to add to what's possible here. W, Y, P. How much damage have we done to our search space? Is this going to take an hour to run? So far, I would say what we've done is not terrible for the search space. M, W, and Y are like the 3 least common amino acids. So adding a mass shift to considering those is not that bad. If we added leucine or valine or isoleucine to our PTM's roster, things would get a lot worse fast because those are very common amino acids by comparison. So searching for a mass shift on an amino acid has more impact on your search time if it's common. These are not. These are all fine, basically. N and Q are pretty common, but ultimately those occurrences happened enough that we need to take them into account. All right, so that's up through oxidation. What are we doing about carbamylation? Carbamylation was pretty common, and helpfully, it appears that it is strongly localized to the N-terminus. It also happens to K, so we should probably include it for both K and the N-terminus. But I think I'm going to leave out aspartate because it's just not that common on the aspartate. The common cases were on lysine and on the N-terminus. So again, let's search for carbamylation in Unimod to get its canonical mass. All right, here it is, 43 point yada yada. All right, and now we go into FragPipe. We're going to add one last mass shift for that. Its mass is this, but we got to get the places it occurs correct here. So it happens on the peptide terminal, but it also happens on a lysine. I want to specify that it happens at either of those possibilities, though. You see, acetylation of any protein N terminal. I got to think about how to do this one. Well, let's start by just we'll add a modification for this happening at the N-terminus of any peptide. So they specify that that should be N-hat, N-caret, basically, like that. And now I'm going to add another possibility for that to happen on lysine. Oops. Make this like the others. 3, lysine, yes. Okay. For some reason, by the way that I ended up replacing things here, you see that there's a maximum occurrences allowable for each PTM. We're allowing for up to 3 individual oxidations on a peptide, only 1 oxidation at a protein N-terminus, sorry, 1 acetylation at a protein N-terminus, up to 3 deamidations within a peptide, up to 3 double oxidations within a peptide, only 1 possible carbamylation at the N-terminus of the peptide, and finally up to 3 if you've got 3 internal lysines, but that's not going to happen because this is trypsin. Okay, does it make sense how I configured these?
You split the N-terminal and the lysine into 2?
I did. If I say lowercase n, uppercase k, I believe the software interprets that to mean on the N terminus of a peptide, but only when the first amino acid is K.
Okay.
That's why I split them.
And you could not use the open bracket column thing that they use for the...
The square bracket means protein N terminus rather than peptide N terminus.
Yeah. Okay.
So you can see that I've been doing this stuff for 20 years. I still get confused at the syntax different people use for this. For me, separating the carbamylations to two separate lines just made sure I was getting what I had tried for. I hope. I mean, we'll see how the search goes. These others are not checked, so they're not live. When we come back to this table, how do we know we're done? I would say you're done adding modifications from the open search to your new search when the explanatory power of the next PTM is too low, that you're not gaining a lot of PSMs for adding the next PTM. The other way to look at it is that you're done adding PTMs to the new search when the search time or the search space becomes too extreme. This is the set of PTMs that we've already listed here are already pretty tough. This search is not going to take forever, but we don't want the search space to balloon out of control. So I think that this is a good place to stop with the set of modifications for now. And the next one is unannotated. We've no idea what chemistry would cause phenylalanine to gain 49. So we can add it, but honestly, I don't care that much about something I can't explain. I'm not going to be investing in a bunch of biochemistry to figure out what happened in those peptides. So I propose this is a good place to stop. And if you want to add another modification to yours, you go for it. Let me know if it finishes, okay? We'll give this a try. Okay, do we need to change anything else? No, not really. Nope. It's all going to be just fine. So I now need to create a new place for my output to go. All right. Training to subsequent search. Once I've done my human interpretation of the open search, I create something that I'm going to call the subsequent search that reflects the bits of knowledge we were able to inject based on the open search. I'm going to close these tables now. All right, and I'm going to close the browser too because I want all my memory, just in case. FragPipe is a bit notorious that when you try to include too many modifications, variable modifications in a search, it can choke and die if you don't have enough RAM. So we're going to try running this as is, and hopefully everything's going to be great. All right, let's do it. Oh, I don't want temporary files. Go away, temporary files. Okay, and now run. Okay, so it could be very quick. It could be very long. What's long? What's too long? I don't know. In the old days, you would start your database search on Sequest on the single server when you left work, and you'd come in the next day, and hopefully everything would be done. Then we got a cluster because people started doing dozens of raw files at a time. These days, these searches can go really blisteringly quickly. I would just say that if we crashed before it said generated blah, blah, blah modified peptides, that would be our indicator that we made our search space too big and the laptop was dying on us. It was just going to say, no way am I going to try this. But in this case, we passed it, so that's really good news. Other search engines do not blow up quite as badly when you ask for a large number of PTMs. For me, one of the better options has been to use MSGF+. It's not a fast search engine particularly, but when you add a whole bunch of PTMs to it, it seems to cooperate with it really well, and it doesn't have the memory required to blow up out of proportion. So it's a good option. All right. Oh, of course, that was the first search, and the first search may not use all of those crazy PTMs. We could still die right here. Let's see what happens. Looking good so far. Did you see that number of peptides with more than 5,000 modification patterns? It's a useful guideline to keep an eye on that. It means that there are some peptide sequences that have so many modifiable locations that there are more than 5,000 possible ways to decorate that one sequence. It's pretty extreme, isn't it? So that's a good way to gauge just how badly things are going for the explosion of your search space as you add PTMs. All right, we're knocking through, doing our validation of PTMs. I think this is going to finish. Looking good. Made it to percolator, right? So how do you interpret something like estimated 7,103 PSMs after a given number of iterations? Is that a good number? Is that a bad number? What's a denominator we can use to compare that to? That's very good, yes. So if it's kind of inarguable that a DDA experiment produces X spectra. You can compute how many tandem mass spectra there are pretty easily. There are lots of quality tools out there that'll do it. I've written some of them. But if you know how many tandem mass spectra you've got, then it becomes possible to say, what fraction of these have I identified, right? So if you have 7,000 spectra, 7,000 PSMs, and you have 12,000 spectra, you should feel pretty good about that. Identifying half your spectra is still a pretty good mark to hit. Is it a bad experiment if you identify 10%?
No.
Not necessarily, yeah. It could be that you're dealing with a biofluid, and in a biofluid, identifying 10% of your spectra is still pretty good. Identifying half your spectra feels better, but that most typically happens when you have mixtures of proteins that are more similar in their concentrations and a great diversity of them. Okay, so our search finished in 4 minutes. Not a problem. What did we find with that? Did we find a large number of modified peptides? Let's check what we got. Okay. Okay. Now I'm accustomed to running this in the LFQ MBR mode. This is a little different here. So where's my peptides list? Here's peptides, I think. So let's try opening that in Excel. Under some modes, it will show you a different file for each modification you've included. Not every time will it do that. So here we go. Great. So we still have assigned and observed. Observed is now a blank column. Okay, so we did not allow for open searching at all here. Only modifications that were on our explicit list are included. So why don't we try sorting our PSMs not by the protein from which they come, but rather by the combination of assigned modifications. Assigned modifications, A to Z, there we go. All right, so we start with some peptides that are deadly dull boring. The 10th cysteine in this sequence has a mass shift of 57 on it. We do not care. That's just the effect of iodoacetamide. So you can see the cysteine in question. It carries 57 mass units. That's lovely. But right away, we start seeing some pretty hefty combinations of PTMs. Now, we told the software we only want to see up to 3 of these PTMs per peptide spectrum match, right? So why are we getting these crazy combinations? The reason is that we have a different limit on each of those PTMs individually. And I don't remember what limit is applied to. I don't remember that there's a limit applied to the peptide in general. Just how many of this PTM are allowed is what we specified. Chris, looking back at our MS Fragger configuration, did we have something where we could set the limit in aggregate? Ah, there it is. All right. Well, so let's do some sleuthing then. What happened here? This looks like more than the allowed limit. Well, this one is a modification, yes, but it is not a variable modification. Cysteine plus 57 is a fixed modification, so it doesn't count towards the ceiling. This is another one. That's also an example of a fixed modification, so that's off the list. But we still have one dioxidation, one oxidation, one deamidation, and one carbamylation. So why did the software produce those? Software gets a little hinky when you start working with PTMs like this. So I'm mystified as to why the limit of 3 was not applied here. All right. So are we seeing any of these modifications that we didn't specify? Anyone see a mass shift that looks unusual? I think these all look very appropriate to me. So yeah, we've got this weird thing going on where it's allowing more modifications than we told it to allow, but on the whole, it looks like the mass shifts it has allowed are the ones that we expected to be applied. Now, it would be a little difficult, if you ask me, to say how popular is any one PTM out of this mixture. So the kinds of plots I'm used to seeing for PTM output would say be a mass shifts on each column and an amino acid on each row, and then I have a count of how many PSMs contain each of these combinations. And a lot of software will produce that. This one doesn't seem to. So that's kind of unfortunate, but it is possible for us to do histograms, for example, on how many of these bore each combination of PTMs. You could do that. It's a little tricky because it has that information about which particular amino acid in the sequence bears that modification. So this is kind of the end of what I've put together for the PTM searching. It would still be valuable to have more information about the localization information here. I wanted to see if we have a probability of localization in here. The probability in column I, I'm pretty sure, is associated with the PSM in general, to say what is the probability that this peptide and this set of PTMs match the spectrum. But I'd like to see information about the localization certainty. And it doesn't seem that this table contains it. That's really unfortunate. That's possible. Oh, yes, of course. Yes, since each of those rows could correspond to multiple PSMs, that may be the answer. All right, PSMs, TSV. Oh, play that chime at me. I love the sound. Okay, spectrum file, peptide, modified peptide. Okay. So we have our mass shifts in kind of a different format here. Sorry?
I think there's a column further out that has the same type of, like if you scroll a bit to like, I believe there is one that's also like assigned modification in the same format.
Okay. Number of miscleavage. There, yeah, that would be under assigned modifications. Yeah, same format, you're right. Yeah. Well, this format of putting the numbers in square brackets is a pretty common way to do it. But in this case, we're seeing that this asparagine instead has a mass of 115. It's not saying there's a mass gain of 115 on asparagine. Does that make sense? So if we see a cysteine in here, Well, the cysteines are being shown as just cysteine because they bear a fixed modification. Their actual mass would be like a C160 instead of a C103. But they only show the variable modifications in the square bracket structure. But I wanted to find localization, so let's dig into that. Whew. This table goes on a bit, doesn't it? Yeah, so the localization is not really featured in this. One of the things you would be expected to do when you publish a paper claiming the recognition of some novel PTM is that you express your certainty in its localization. And simply working with the spectrum to show how the score drops as you move it from position to position is one way to go about that. In this case, the PTMs we had were all pretty tightly associated with a small group of amino acids, except for single oxidation. We allowed 4 different amino acids on which oxidation could occur. And we're not really getting information here on how sure we are that it's this amino acid and not that other one. Okay. Well, the other thing I guess I would point to at the end of the day is this protein description stuff. Do you see what proteins show up here? Beta crystallins, alpha crystallins. Anyone know about the role of crystallins?
Yes, this is from eye lens, right?
This came from a human eye lens for a person who's 93 years old, no cataracts.
Okay, well done.
I think what this tells us is that the major proteins that refract light in our eyeballs are precious. We should protect them. So don't stare at the sun and take good care of yourself. Eat things with antioxidants in them because your body gets exposed to a lot of stuff and it does degrade the proteins that it comes into exposure with. I, for one, will be wearing sunscreen when I'm next on a beach.
Also for other reasons, but
yeah. Yes. All right. Are there any other questions about PTM hunting? Well, I really appreciate your taking some time with me on a Friday afternoon. Chris, you had a thing to say.
I was wondering, so there is a separate workflow for glycoproteomics.
Yes.
But what would the feasibility be of doing an open search that includes modifications up to large glycans, which can be like thousands of golden dark?
I know one person who has attempted to use open searching for cross-linked peptides.
Yeah.
That is a hard case.
So then your modification is essentially an entire extra peptide.
That's correct.
Ouch.
Yes. You can abuse the feature,
Yeah.
and it may work well for you.
But it sounds like it's kind of
Ultimately, we would like to think that cross-link identification software is better at interpreting such spectra. Occasionally, it's good to use bog standard tools like FragPipe for doing basic sanity checks on the data. And an open search is not the worst way to accommodate the fact that you've got cross-linked peptides in the mix. When you do a cross-linking experiment, it's still very frequently the case that the great majority of the spectra you have represent either loop link or unlinked peptides. So the bulk of your data will still be identifiable by a conventional search engine, quite apart from the cross-link ones. So yes, you could use OpenSearch as a way to go after glycans of unknown size.
But you will end up with like a pretty massive search base, I guess.
It doesn't pain things a lot to do that. Once you allow for open modifications in a range of 650, I think, Daltons is what we had in this case.
-150 to 500.
Yeah.
So the glyco aspect of this would use like libraries of glycans, and they range in size from like each like like a mannose, for instance, weighs like 167-ish.
Yeah.
And then up to 20 times that.
Yes.
And so you're looking at a search space of 100 to
the worst possible search space for open search is that every peptide gets compared to every MSMS.
Yeah.
But you're already reasonably close to that using a search space of -150 to 500. It's not going to change tremendously by upping the ceiling to 2000.
Yes, you're still limited in the number of sequences you actually have to work with and number of spectrums that you have.
Yeah. Especially if you're dealing with a subset database that contains only a few hundred sequences. It will run, and it will probably give you a reasonably sensible response as well. I would say that the bigger problem is that when people do PTM hunting, they do so with the assertion that having a PTM just changes the mass ladder. But the reality is a lot of PTMs change how the peptide fragments. And that's the problem. So let's imagine that you've got a big glycan structure stuck on your peptide. And that glycan structure breaking away from the peptide is more labile than the peptide bonds. In that case, you get a big loss ion and no fragments. So I would say that's the bigger question. How does a modification of that magnitude alter the fragmentation of the peptide overall? If it stands in the way of the formation of B and Y ions, you're going to have a problem. Yeah. Okay. Thank you for spending your Friday with me. I hope that you get to enjoy the GJAM meeting as well, if you're going to go. Yes? Good. Awesome. And thank you for letting me walk through this presentation in advance. I think this will help me have a more coherent presentation for Tuesday. So thank you very much. Great. Thanks.